So welcome to elevator pitch 10 for, or the elevator pitch session number 10 for the STEM section of the Polygens Symposium of Rising Scholars. My name is Gokul. I'm a PhD student at UCSF, the University of California at San Francisco, and I will be your moderator for today's session. So the goal of the symposium, and this is the 7th Polygen Symposium, is to celebrate and showcase the hard work of our Polygen scholars, all of whom have worked tirelessly on their projects over the past couple of months. We have 7 students presenting on a variety of topics today, and a fellow member of the Polygen team will be scoring each student's presentation after the event, which will help us determine the prize winners. So if you have any questions for the speakers, or if any audience members also trail in over the course of the seminar, please feel free to type your questions in the Q&A section on the Zoom down below. If you have any questions for each speaker, and then at the end of the session, after all the speakers have gone, we'll have some time for everybody to ask those questions, or maybe I'll read out those Q&A questions out loud to the appropriate speakers. Okay, so we decided that the order of presenters will be in alphabetical order by first name. So I think our first student is going to be Akhil. All right, so go ahead and you can start, and then everyone else, we can stop our video and mute ourselves, and then I'll turn my camera back on when you have 1 minute left, just as a note to start wrapping up. Sound good?
Yeah, thank you so much. All right. Well, hi everyone, my name is Akhil Rajidi, and this presentation is going to be about investigating the role of microglia in Alzheimer's disease. So first, we're going to discuss what microglia actually are, and then we're going to take a look at Alzheimer's disease and how microglia impact its pathology. And then finally, I will conclude with some positive news on therapeutic approaches scientists are investigating in microglia to help with Alzheimer's disease. So what exactly is microglia? Microglia are glial cells that act as the immune cells of the central nervous system. These specialized cells serve 3 main functions: sensation of changes in the environment, phagocytosis to promote neuronal well-being, and a defensive function in response to injurious changes. Microglia are continuously active, sensing the environment for any changes to make an informed decision on whether to perform their housekeeping or defensive actions. For example, these cells can take part in the pruning and remodeling of synapses or phagocytosis of dead or dying cells. They can also initiate neuroinflammation in response to infectious pathogens by producing cytokines, which summon additional cells to get rid of the infectious agents. So let's take a look at Alzheimer's disease. Alzheimer's disease is a disorder that is associated with impaired memory and important mental functions that can lead to a loss of thinking skills and the ability to carry out simple tasks. You may be familiar with its main symptom of dementia, but it can also cause other symptoms such as unfounded emotions, lack of social contact, and a difficulty with speech and judgment. Alzheimer's disease is characterized by the accumulation of amyloid beta and severe cellular damage, and has been associated with certain mutations for decades. Despite the involvement of these general and known gene mutations, the early stages of the progression of pathology can't be explained simply by these mutations, suggesting that there must be another process in AD pathology that must be activated to trigger neurodegenerative disease, which is why I would like to turn our attention to recent research on microglia. Some genes specifically expressed in microglia include ApoE, TREM2, and CD33, which are highly linked to neurodegenerative diseases. All these factors are thought to exacerbate AD due to their involvement in amyloid beta and amyloid beta clearance. Modulating these genes has been shown to have beneficial effects in a range of mouse models. This altogether suggests that microglia have a role in AD, including some evidence that it could be driving the initiation or progression of the disease. This makes microglia all the more important as a target for researchers. Due to the fact that microglia activation may drive disease progression, researchers have pursued microglia depletion approaches followed by repopulation by residual microglia. This has led to beneficial effects in AD mouse models where the microglia were eliminated by inhibitors of CSF1R, which is a microglia receptor that's important for signaling. This improved cognition and reduced neuroinflammation. However, further research needs to be done on the specific microglia subpopulations and how they impact the central nervous system at different stages of the disease. We can also take a look at inhibiting specific pathways that promote inflammation in the brain. The NLRP3 inflammasome pathway creates protein complexes that activate caspase-1, which ultimately triggers a cell death pathway. In mouse models, the activation of this pathway has shown to cause synaptic dysfunction and cognitive effects. Deleting the gene for NLRP3 has been shown to have protective effects in disease models. Major drug development efforts have targeted the NLRP3. However, NLRP3 inhibitors still need to be tested in clinical trials for their safety and efficacy. So in conclusion, neurodegenerative diseases impact the lives of 1 billion people across the world, which motivated me to write my paper not only on microglia and Alzheimer's disease, but also Parkinson's and ALS. We can better have a grasp of the underlying causes of these diseases by looking at mutations that occur in the biological process of the brain. It is interesting to see that many of the genes or mechanisms found to have an impact on pathology have been excessively or exclusively expressed in microglia. Hopefully, future research can really take advantage of these findings to find some therapeutic approaches. I just want to give a shout out to my mentor, Dr. Kevin Guttenplan, and here are my references, and let me know if you have any questions.
Awesome. Thanks so much, Akhil. And again, for any attendees who are watching it live, obviously this is recorded for everyone else, but people who are watching live get the opportunity to ask any questions. You can feel free to ask them at any time. Just type them into the Q&A tab down below on Zoom, and then we'll address them all at the end for all of the speakers together. Thanks. So our next speaker is Matthew. Do you want to turn your video on and get started?
Yeah, sure. Let me share my screen.
Perfect.
Give me a second. Okay. Hi, my name is Matthew, and today I'll be talking about my review article about the applications of tissue engineering in cardiac cells. So my overall goal with this paper was to better my understanding about the cardiac field and tissue engineering in general, in order to learn about the current state of bioengineering and fulfilling my interest about learning more about this field. So currently, cardiovascular disease is the leading cause of death, and some remedies that are currently available include non-invasive and invasive techniques that accomplish lowering blood pressure, heart rate, and overall just ensuring that the proper function of the heart is stable. Cardiovascular disease accounts for 1/5 of all deaths worldwide, and 700,000 people died from cardiac disease in the United States alone in 2020. With cardiovascular disease, the cells of the heart are not functioning properly and need extra assistance for survival. In these cases, heart transplants are common. However, they're in a lot of shortage because it takes a long process to find a match and make sure that it's functional. A solution in the works is cardiac tissue engineering, which is a process that involves bioengineering cells in order to sustain proper function in the heart. It involves isolating healthy cells from the body and culturing it outside, and afterwards implementing it back into the body, which will help for cell function for desirable cells. The current state of tissue engineering involves bioprinting, stem cell injections, cell sheets, and decellularization. Bioprinting is very similar to 3D printing, but it prints organs in either parts or wholes by stacking cells on top of each other. Stem cell injections involve injecting cultured cells into the body by using hydrogel or other scaffolding techniques to aid in that process of injection into the body and also encourage cell growth in the injected area. Cell sheets are structure created purely out of cells and are surgically implemented like a transplant. And finally, decellularization is a process that extracts cells from a healthy extracellular matrix, or in other words, a healthy cell environment in the body, and replacing it with desirable functioning cells to improve cell function in that area. Some current challenges in tissue engineering is an overall lack of testing. Tissue engineering is a very new field, and the finer details of the 4 strategies I listed before need to be worked out in order to have a successful implementation in the medical field. Some plans for the future involve working out these finer tuned details, such as printing at a smaller resolution, finding the perfect duration for a hydrogel to function, understanding the properties of cells and determining new techniques that may work better than the current ones. And despite all of the improvements that can be made, tissue engineering shows a lot of promise in advancing the cardiac cell treatment process and has a very strong foundation that can be perfected over time in order to solve the significant problem of heart disease. Thank you.
All right, thanks, Matthew. And again, for everyone, feel free to ask any questions if you want. You can put them in the Q&A, but we'll get to them after all of the speakers are done. We want to answer them one by one. Awesome. So continuing down the list of alphabetical order, I think our next speaker is Rachana.
Yep, let me share my screen.
Sure. Spotlight you.
Okay. Can everyone see my screen?
All right, take it away.
Hi, everyone. My name is Rachana Gurudu, and today I'll be presenting my research paper on a machine learning model to predict diagnosis and prognosis in patients with coronary artery disease and myocardial infarction, as well as the identification of biological pathways, drugs, and immune cells, also related to CAD and MI, to improve biological understanding of the conditions. To begin with a background, coronary artery disease, or CAD, is the leading cause of mortality in the United States, with over 3 million people diagnosed annually. CAD is caused by atherosclerosis, or arterial plaque buildup, and can eventually cause cardiac ischemia and myocardial infarction, which is a heart attack. Although certain genes have been found to be significantly correlated with CAD and MI, finding consistent and replicable genetic markers is key to finding patient and gene-specific treatments for this disease. Additionally, existing research on gene expression data in CAD and MI patients is often not heterogeneous, causing machine learning models to be inaccurate for all populations and demographics. This study uses meta-analysis of datasets globally to increase biological heterogeneity and find more accurate genetic markers. For our study's methods, we obtained 12 datasets from the NCBI Gene Expression Omnibus, which is a source from the NIH. To create our gene signatures, we used the R meta integrator package, which performs statistical meta-analysis on all datasets and identified significant genes. Next, we used the Library of Integrated Network-Based Cellular Signatures, or LINCS database, to perform drug analysis on the gene signature. And then we also performed immune cell deconvolution on our gene signatures, which allowed us to identify significant immune cells in CAD and MI patients. Lastly, we performed biological pathway analysis using Reactome, an online collection of biological pathways to identify significant pathways associated with both diseases. For our results, we divided our results into CAD versus healthy patient datasets and CAD versus MI patient datasets, and used forward search algorithms to identify significant genes. The CAD versus MI gene signature was highly predictive on our discovery datasets, as you can see in the plots on the right, with the areas under the curve, or AUCs, being greater than 0.8. The CAD versus healthy gene signature was less predictive and had lower AUCs. We then performed an immune cell deconvolution algorithm on both the CAD versus healthy and CAD versus MI gene signatures, and found B cells, granulocytes, neutrophils, and natural killer cells to be significant. For our drug analysis, we identified nifedipine and myriocin as significant drugs for CAD patients, and enzalutamide and ibutylide for MI patients. These drugs are all indicated for other purposes like hypertension or prostate cancer, but showed promising effects towards reducing the effects of CAD and MI. This plot shows the outcomes of our biological pathway analysis. As seen here, the brighter dots indicate the more significant correlation with our gene signature. The two most important outcomes here are oncogene-induced senescence, which is a pathway associated with tumor suppression, and neutrophil degranulation, which is the release of the white blood cells neutrophils. Creating this machine learning model predictive of CAD and MI in patients allowed for increased biological heterogeneity in our gene signature, as well as the identification of more accurate drugs that can be repurposed for treatment or prevention, and immune cells that have a significant relationship with CAD and MI. However, the study has some limitations. First, data across data sets was collected at different time points following patient diagnosis and treatment, which may have affected gene expression, and future studies with a more standardized collection process would improve the predictive levels of the model. Second, limited data exists on the relationship between the drugs and pathways we identified, and future studies are necessary to examine these relationships beyond initial computational correlations. Thank you.
Great, thanks. Great, you guys have all been perfect on time so far, so great job. All right, next is Ruhika.
Hi everyone, my name is Ruhika Muralidhar and I worked with my mentor Joseph to analyze the use of stem cell therapies for lung cancer patients with a history of tobacco use. Lung cancer is caused by a malignant tumor in the bronchi, bronchioles, or alveolus of the lungs. There are two main types of lung cancer: small cell carcinoma, which is more common and severe, and non-small cell carcinoma, which is less aggressive. In this study, we focus more on the treatment of small cell carcinoma. Lung cancer makes up about 25% of all the deaths that are caused by cancer, with the majority of these deaths coming from smokers. It is the most frequently diagnosed cancer in both men and women outside of breast and prostate cancer. After diagnosis, only 40% of patients are able to survive for a year or more, and only 15% of patients are able to survive for 5 years or more. The leading cause of lung cancer is smoking. Inside of a single cigarette, there are about 7,000 chemicals, with 70 of them being carcinogens. Carcinogens are cancer-causing agents that can lead to damage the cells and lead to mutations. This is why it is crucial to avoid smoking to eliminate the possibility of developing lung cancer in the future. Some examples of the carcinogens are shown on the slide. There are currently 3 different kinds of treatment methods for lung cancer: chemotherapy, surgery, and radiation therapy. While they have some benefits, the current treatment methods do not always treat patients in the long run, and they do need some advancements. One possible treatment method for lung cancer is stem cell therapy. Stem cells are undifferentiated cells that can be used to build tissues in the body. There are 2 kinds of stem cells. Embryonic stem cells have the ability to differentiate into any cell in the body, while adult stem cells have specific functions they'll end up performing. Because of the ethical concerns of using embryonic stem cells for treatments, scientists discovered a method to genetically modify adult stem cells into embryonic stem cells. There are different types of stem cells in the body that can be used for treatment. In most studies, scientists choose to use mesenchymal stem cells as they are found in the bone marrow for easy retrieval and return back to the body. Once the stem cells have undergone their modification process, doctors commonly use 3 types of methods to return them back into the body: local injections, in vitro scaffolds, and tissue-engineered scaffolds. Local injections contain a 3-step process shown above to help the patients get comfortable with the stem cells in their body. In one study conducted by Hijisem et al., scientists tested the benefits of local injections on patients with hepatocellular carcinoma and discovered that stem cell therapy showcased major benefits on patients when combined with chemotherapy. Another study conducted by Dalal et al. Discussed the benefits of local injections on patients with Crohn's disease, and they were able to notice a reduction in the size of the lesions formed by the disease. Both of these studies are able to show the benefits of stem cell therapy on lung cancer patients because they were able to treat severe problems on vital organs and regain their function. Future testing can be done to prove this theory. Another delivery method is scaffolding. A scaffold is a medical technique that forms a framework that creates an environment to facilitate the formation of desired tissue. There are two kinds of scaffold: in vitro scaffolds, which are conducted on a petri dish, and tissue engineered scaffolds, which are designed using collagen or polylactic acid. One study conducted by Hawley et al. Explains the general pattern of growth that is experienced with the treatment of cancers using in vitro models. In another study conducted by Luo et al., scientists have analyzed bifunctional scaffolds as a potential treatment for breast cancer. Both of these papers show proof of the potential use of stem cell therapy for treatment, as they showcase how the scaffolding method has been able to resemble tissue in the body and help with its growth. To summarize, the first and most beneficial way to avoid lung cancer is to avoid smoking. Quitting smoking and the use of other tobacco products can help prevent carcinogens from entering the body and help avoid lung cancer in the long run. If it is too late and treatment is needed for small cell carcinoma, then stem cell therapy is shown to be an extremely beneficial treatment method for this disease. Along with stem cell therapy, chemotherapy can be paired in order to form the best possible treatment method. The chemotherapy can help diminish the tumor and prevent growth of cancer cells, while stem cell therapy can help initiate the development of healthy cells to quicken the healing process. This result can be used for future research to see if chemotherapy paired with stem cell therapy can be a newfound treatment for lung cancer. Thank you for listening to this presentation, and here are all my sources for the research papers.
Awesome, great job, thank you. All right, so I think next is Sanjana.
Yeah, let me share my screen.
Mm-hmm. Oops.
Okay, hi, my name is Sanjana, and my literature review is about sleep, the circadian rhythm, and mental disorders or illnesses. So what is the circadian rhythm? The circadian rhythm functions as the body's internal clock to regulate periods.
Can you wait 1 second, sir? At least on my screen, it shows it's not showing your slides. It just says loading.
Oh,
Sorry to interrupt.
let me try sharing it again.
Yeah, no worries. Take your time.
Are you able to see it now?
Yep.
Okay.
Oh, now it's back to saying loading though.
Oh.
Oh. It seems like it's an issue with the present button.
Yeah.
Maybe you should click another way.
Yeah, give me 1 second.
Yeah, for sure. We're doing great on time, so don't worry. There's no rush.
Okay, I will leave it in this mode so that it's visible. Can you see it now?
Yeah.
Okay, my name is Sanjana, and my literature review is about sleep, the circadian rhythm, and mental disorders or illnesses. The circadian rhythm functions as the body's internal clock to regulate periods of rest and activity over a 24-hour period. Sleep, the rest portion of the clock, is crucial for maintaining the balance of the circadian rhythm. An imbalance in the rhythm is known to disrupt many bodily functions such as hormone release and immune response. Conversely, many mental disorders are caused by an irregular pattern of hormone or neurotransmitter release. What are some mental disorders or illnesses associated with the circadian rhythm? My research aims to find effects of sleep on the balance or imbalance of the circadian rhythm that makes an individual more prone to developing mental disorders such as anxiety, depression, or ADHD. Why is this important? Finding a correlation between sleep and the development of mental disorders would allow researchers to find possible treatments by modulating sleep. Currently, the main forms of treatment for mental disorders such as depression are therapy and pharmaceuticals that target neurotransmitter release in the brain. Alternative treatments could arise from studying sleep in patients with mental disorders. What are some possible treatments? The first possible treatment is sleep therapy. Medications such as melatonin and CBD allow a doctor to have some level of control over the patient's sleep cycle, allowing for the regulation of sleep and wake times and consequently the length of the rest cycle. Melatonin and CBD, among other sleep medications, activate a gland in the brain that triggers feelings of drowsiness or fatigue, which encourage the patient to fall asleep, initiating this rest cycle. The rest cycle is where tissues are regenerated and hormones are regulated and balanced. So regulating sleep allows for the re-regulation of these hormones and neurotransmitters such as serotonin, oxytocin, dopamine, and melatonin. In some cases, sleep therapy is not sufficient and light therapy is a more promising form of treatment. Generally, the human body sets active and rest periods by taking into account the daily amount and timing of ambient light, exposure to specific periods of light and dark cycles, which varies from treatment to treatment, can change the body's release of molecules that dictate rest and activity. Ultimately, light therapy could yield the same results as controlling the activities to sleep, which is sleep therapy. This diagram outlines how sleep therapy works, essentially resetting the rest portion of the circadian cycle over time allows for the restoration of neurotransmitter regulation, which in turn suppresses the expression of certain disorders. And this diagram outlines how light therapy works. Instead of directly controlling the rest portion, a light adjustment factor is introduced, which changes the body's perception of night and day at a molecular level and restores neurotransmitter regulation. Further research is needed to determine a more definite and reliable method, but for now, sleep and light therapy seem to be the most feasible non-invasive treatments. Thank you.
Alrighty, thank you. And good job problem solving with those slides as well.
Yeah, no problem.
Awesome. So I think next is Skylar, is that right?
Yeah, thank you.
Mhm.
Let me share my screen. Okay, so hello everyone. My name is Skylar and I'm currently researching active targeting nanocarriers to be used as cancer drugs. So first of all, why do we need this new form of drug delivery in the first place? Let's first start off with addressing the issues of current cancer therapies. So here below are listed some of the most common forms of traditional cancer therapies, and each have their own limitations, leaving lots of room for improvement to fight back. As you can see, their main problems mostly stem from the fact that they harm the healthy cells or they're unable to target the specific tumor, as many cancer tumors spread into all sorts of interesting shapes, creating a vague line between the cancer tumor and healthy cells, so they're generally unpractical. For example, for the ones that are unpractical, current targeted treatments such as Gleevec, they basically consist of proteins that only bind to certain mutations, but it means that there are also limitations to only specific mutations, and immunotherapy in addition also is very expensive and also have that specific requirement. So going back to my topic of research, nanocarriers are essentially very tiny packets that encase a little bit of cancer drugs in them. And even though each nanocarrier can only carry a little bit of cancer drug, with a bunch of them, you can have a lot more cancer drugs. So what's the point of having such a small size? And why wouldn't we need to encapsulate them? Well, the whole point of nanocarriers are so that they can overcome the limitations mentioned in the previous slide. I'll start off with how nanocarriers work. So in passive targeting, there's two kinds of ways to use nanocarriers, passive targeting and active targeting. So in passive targeting, nanocarriers are injected into the body near where the tumor is located, and they flow through the bloodstream and hopefully into the tumor site. And this is dependent on what is called the EPR effect, also known as the enhanced permeability and retention effect, which states that fast-growing tumors tend to grow messily, causing leaky blood vessels, giving the perfect space for nanocarriers to flow in. In addition, in active targeting, the only difference is that instead of hoping that the nanocarriers can reach the tumors through luck, scientists attach a targeting mechanism onto the nanocarriers, which can help actively direct them into the tumor. And after reaching the tumor cells, they can then become engulfed by the cell and finally release the drugs through methods such like biological stimuli or light activation. So the advantages of using nanocarriers is that due to its size, it can overcome many biological barriers with ease and also have that one-on-one interaction with the targeted parts instead of promiscuous interactions with other areas that result in affecting healthy cells, which occurs in current therapies such as chemotherapy and radiation. In addition, they also do not require the tumor to be solid like, for example, surgery, since there are many nanocarriers and they can all go their separate ways into the different parts of the tumor. So an important aspect to mention, however, is that even though their nanocarriers have great advantages, there are also many limitations to it. Because nanoparticle and nanotechnology were only recently developed and researched upon, there's not enough research to declare them as safe. So currently there are only a few nanoparticles that are FDA approved. In addition to the overall safeness of the nanocarriers, we also have very few tests of how the nanocarriers act in the human body. So as most of the tests are done in petri dishes, which poorly resemble the human body and emit many important factors, such as the biological barriers. So even though nanocarriers have great potential, these issues need to be resolved first before they can actually reach the market. Okay, thank you for listening to our presentation.
Great. Thank you, Skylar. And I think our last presenter is Vivek, is that right?
Yeah. I'm sorry, I don't know what's going on with my background, but something wrong with maybe the lighting right from the window. But I'll go ahead and share my screen.
Sure.
Okay, can you guys see? Is it all good?
Yep.
Okay. So hi guys, my name is Vivek Nalluri, and today I'll present my research review on acute lymphoblastic leukemia and its genetic association within different ethnic cohorts. So acute lymphoblastic leukemia, abbreviated with ALL, is a cancer found in the blood and bone marrow. It creates immature white blood cells called lymphoblasts that crowd out mature white blood cells. So this lack of mature white blood cells, which is what ALL is, leads to a weakened immune system. So ALL is diagnosed most frequently in children under the age of 18, accounting for more than 30% of all childhood cancers. The graphs on the left show how Black, Hispanic, and white populations have the lowest survival rates, while Asian populations have the highest. So during COVID, one of my mutual friends passed away from leukemia. It had made me realize that kids are not invincible, contradicting the common stereotype of us being healthy. So I pursued research on childhood leukemia to learn a little bit more about this common disease. So we have already established the racial disparity of ALL survival. And in addition, childhood ALL was found to be 20% more common in Latino American populations compared to non-Latino white populations. What is not known about ALL and cohorts of various ethnicities are the patterns of which common genes expressed within those different populations are affected by ALL. So my research statement was that through literature review, I investigated the common genes that are expressed in various ethnic cohorts affected by ALL and the mechanisms of upregulation of these said genes. So my research has identified 3 main genes that are associated with ALL. Variations of these three genes called polymorphisms are correlated with increased susceptibility to all. Polymorphisms of XRCC3 THR241 MET were initially shown to be more prevalent within the Asian population, but then discovered to only be correlated with the Caucasian population. And polymorphisms of the ARID5B gene were noted to be more frequent in Hispanics, potentially explaining their higher susceptibility to all. And lastly, variations of the FOXO3 gene were identified to be associated with increased risk of childhood all, more predominantly with the Chinese population. So polymorphisms of these three genes are associated with an increased risk of acute lymphoblastic leukemia within different ethnicities. XRCC3 THR241 MET, ARID5B, and FOXO3 polymorphisms are also associated with other cancers, including breast, prostate, and gastric cancer, which further validates my results and implicates their role in cancer susceptibility. So currently, there are not many targeted drug therapies for all, and none of them are specifically suited for the polymorphisms I studied. My review on these genomic locations marks them as potential biomarkers for targeted drug therapies for all. So further study on these genes could surface groundbreaking information in targeting and treating children with all, so that we reduce the fatalities and the susceptibility of all within our world. So these are the citations I've used. I have quite a bit of researches that I used to look at different studies and literatures. And yeah, I hope you guys have learned a little bit something about acute lymphoblastic leukemia today. Thanks for listening.
All right, thanks everyone. And Vivek, would you mind ending this? Yeah, perfect. Thank you. Yeah, thanks everyone so much. And great job on staying on time as well. I know it's probably really hard summarizing such an impressive amount of research in like 3 to 5 minutes, but you all did a perfect job of staying on time and doing that in a way that captures all the main points of your research, but also doing it in a format that gets the point across really quickly and in a way that other people can understand outside of your field as well. So I actually had, since we have a couple extra minutes and we finish early, I had a couple of questions that I wanted to ask all of you, and I'll just ask both at the same time. Maybe you can go through in the same order and you guys can give your answers. The two questions that I had are, one, these are some very advanced research topics. As someone who's doing a PhD in a biological field, right? Like these are the kinds of questions that I would see like my colleagues maybe giving presentations on and summarizing, right? So what is it in your experience that made you want to pick your topic as something you wanted to study? I think Vivek, you mentioned a personal experience that made you want to study this ALL in particular, but for the rest of you as well, I'm very interested in hearing, first of all, what made you interested in this field of research? And the second question is, if you could tell the field, one statement of the conclusion of based on the conclusion of your findings, like what a scientist in your field should do or should look into, for example, what would that one sentence takeaway be that you would give them? And maybe we can go through in the same order.
Yeah, sure. So one thing that kind of prompted me to follow this research topic. So initially I was coming into Polygens, I was already interested in studying something about neuroscience. But after talking with my mentor, to specify a topic, I became more interested in cells that neurons, not because there are a bunch of other types of cells in the brain that aren't just neurons. And I wanted to study, specifically in terms of neurodegenerative disease, because I know that the neurodegenerative diseases are some of the hardest to treat, some of the hardest, they're not even curable yet. So I found that very interesting to take a topic that I don't really know about yet, which is cells that are not neurons, and also study it in a context which I'm really passionate about. And if I were to tell what future research should be on, I would say if scientists were able to focus more on microglia or any types of cells that have specific genes that they know can trigger a disease, like maybe focus on these cells as a therapeutic, like towards developing a therapeutic approach. And yeah.
My initial project was actually working towards tissue engineering in the food industry. I watched a super interesting video about someone creating artificial salmon through a bioreactor. And my initial idea with polygens was going through with that. And then as I was researching, I realized that there wasn't much information out there, but I still wanted to have the same passion for tissue engineering into a different field. And I was going through different like problems that are happening in the world, and with cardiac problems happening so frequently in the world, I thought that's a good point to target and a good point to research on. So I have more information about tissue engineering and also overall about like the biology of the human body. And something that I would prompt other researchers to follow is implementing tissue engineering inside of human patients, because right now it's a lot of testing in animal models and testing in vitro. So something that could be improved is implementing it into humans and see what problems arise there and trying to fix them, so we can build a bridge between the research and actual implementations in humans.
Okay, I chose my research project, which was creating a machine learning model to identify CAD and MI, because I'm very interested in the intersections of like computer science and bioinformatics and their applications on medicine. And I'm also really interested in diversity and equity. And I think that this project was able to incorporate aspects of that by using data sets from around the world and create a more representative gene signature, which was my goal. I also chose coronary artery disease because I have some family members who have struggled with that in the past and wanted to further the research in that field to hopefully find new applications of treatments. And then for the second question, I think I highlighted this at the end of my presentation, but I think having more standardized collection measures would allow for a more predictive model, which may help patients in the future. So collecting data on CAD and MI patients at specific time points, and also incorporating more diversity in research studies related to these diseases to allow for a better representative model and more accurate treatments.
I chose my research topic for a variety of reasons. So one of them was being that a lot of people I'm close to and a lot of people I'm related to, and like friends and family, they all have had experience with tobacco products and smoking. And so I wanted to be able to integrate that into what I wanted to research, because at my school, I've been more interested in spreading awareness about this topic. So I thought being able to expand more about how it impacts certain diseases and how it can be treated, I thought it was very interesting. And then when I was looking back, when I was in middle school, I always found the idea of cancer research to be very interesting overall. So I kind of wanted to involve how I can treat cancer, because lung cancer is one of the problems that are caused by smoking, and along with emphysema and other diseases. But that was something I wanted to integrate. So I kind of connected smoking with lung cancer to be able to analyze that. And then after that, when I was a freshman, I learned a lot more about stem cell therapy and how it can be beneficial for cancer and other regenerative diseases. So I thought if I combine all these ideas together, I would be able to learn more about all three of these topics while figuring out a solution in the best way I could as being a high school student. And then one thing I would like to tell scientists based on my research is that to be more open to the idea of using stem cell therapy with other treatment methods for more severe forms of cancer, as right now what I've seen in my research is they've used it for more like skin cancer and more like localized cancers, while lung cancer is a very common cancer, but it can be very beneficial using stem cell treatment methods. So that's what I probably would want to tell.
My interest in neuroscience started with taking AP biology, And my teacher really had a unit that focused on it. And since then, I started being interested in more of the biological aspects of neuroscience and cognitive science. And I decided to focus on lack of sleep for this research, mainly because it's such a common problem, especially with people our age. And I wanted to know more about the effects of lack of sleep and how widespread it actually is. And for future research, I would propose researching more about sleep therapy, because it's there as a treatment right now, but it's a very vague term. There's not much known on the details of the process or how it actually works. So I think knowing more about how you can standardize it would increase how effective it is.
So I'm actually more interested in being like a physics major and that side of science, but I got into Marvel and I was inspired by Tony Stark's nanotechnology, and I researched and how he incorporated it in that suit. So I researched like the applications of nanotechnology in real life, and I got into like learning about nanoparticles and like, for example, quantum dots, and then they use quantum dots to diagnose cancer. But I thought it wouldn't like, but I thought it would be more useful to actually be able to like get have a way of using the therapy. So then I came across nanocarriers. So I guess utilizing this technology on the nanoscale and putting it into practicality to save lives is what really drew me in. And something that I would prompt researchers is just for them to stay in this field of study of nanocarriers, because I know the current results aren't really impressive, but I believe like if more research is done with targeting and testing with the different kinds of targeting ligands and mixing and matching between the properties of these nanocarriers, I think there's a lot of potential in this kind of therapy. Yeah.
So the reason I decided to pursue my research on acute lymphoblastic leukemia was, like I said, the personal experience I've had with it. But also it was, I would love working with kids, and, I have a tutoring business, and it's just, kids are so like, I enjoy spending my time with kids, and I think, they're so like, it's, it's hard to explain, but I enjoy working with kids. And also the field of oncology has always been something that interested me and pulled me. The fact that you just can't cure cancer, right? It's so diverse, and so in particular, you just can't cure it, right? So that's something that pulled me towards it, and I decided to pursue my research project on something that combined both of them. Leukemia is the most common childhood cancer, so I decided to dive deeper into ALL. And one thing I would say is that, in science, nothing's ever been proven right. It just hasn't been proven wrong. So I think that's something good to keep in mind when looking at, especially a field like cancer, because it's so open-ended. You just have to keep looking. You have to go in with an open mind. And in terms of my research, it's better the bigger studies you have, the better. If we could get a study with every single leukemia patient in the world, you'd probably have the strongest results there. But obviously, that might not be physically possible. But so, the larger study you can have, the better it would be to get the most, strong results. So going forward, in my research, I chose, I tried to make sure I could have the largest studies I could find. And, there were obviously like contradicting statements within the literatures I was looking at too. One literature said, it was only more prevalent in an Asian population, while another literature on the same polymorphism was like, no, that's completely wrong. There's no association between the Asian population, but there is an association with the Caucasian population. And, that study had a wider and more, bigger study group. So, it's just, have to keep an open mind with cancer, and, not really don't be afraid to find false results, or even, not accurate results, because it can always be proven wrong.
Thanks everyone for sharing. I think that's it's really inspiring for me to see that the world is in good hands, because I think you guys are doing great work already, and I'm excited to see what you do in the future. So yeah, with that, I think we can end the session early. Feel free to hop into some of the other sessions that are going on around now. I think some of them had more students than this session did, so they might still be going on. So you might catch a cool talk at the end of one of the others. So yeah, have fun, everyone.